Hello, everyone, and welcome to today's webinar, High Content Phenotypic Screening, Answering Questions You Haven't Even Asked Yet. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Molecular Devices. To learn more, visit moleculardevices.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Angeline Lim, Senior Application Scientist at Molecular Devices, and Dr. Victor Wong, Chief Scientific Officer at Core Life Analytics. Angeline and Victor, you may now begin your presentation.
Hello, and welcome to the webinar. My name is Angeline Lim, and I'm a Senior Imaging Application Scientist at Molecular Devices. Today's webinar is divided into two parts, and I will start off with an introduction to high-content phenotypic assay, specifically focused on cell painting. Now, because such high-content assays tend to generate lots of data, in the second half of the webinar, Victor Wong from Core Life Analytics will tell us how we can work with such big data. So to get things started, I'm going to kick off this presentation with an introduction to cell painting. So what is cell painting? This is a phenotypic image-based profiling method that has widespread applications in drug discovery and also in functional genomic studies. It was initially developed and optimized at the Broad Institute and in Carpenter's lab. And the aim of cell painting is to visually represent as much of the cell as possible. And so to do this assay is therefore highly multiplexed. Cells are stained with multiple fluorescent dyes. And for cell painting, you would do that with up to 6 fluorescent dyes to label various compartments in the cell. The images are then used to generate an information-rich morphological profile which characterizes each cell. Now, I would also like to take some time to explain the difference between phenotypic screening versus phenotypic profiling, just to give an idea of how powerful profiling is. So, with screening, you would typically quantify a few features. And these features are selected based on some known association with the biology. When it comes to profiling, the idea is to quantify everything. And so all the measurements that you gather form a so-called fingerprint that represents the cell biological state. And as I've heard before in other talks, there's someone who said, in profiling, you would measure everything and ask questions later. And so here's a simple illustration that shows compounds A and B have a similar phenotypic fingerprint and are both quite different from the effects of compound C on cells. In addition, profiling is also an unbiased approach. And when you take an unbiased approach in your experiment, it increases the possibility of making some novel discoveries since it is not limited by some prior knowledge. It is also more efficient because a single experiment can be mined for other questions that you may have later. And so let's say, for example, that in the last couple of slides, I've gotten everyone in the audience very excited about cell painting and you just want to get started. So how does one go about carrying out cell painting? So in a standard cell painting assay, cells are perturbed using chemical or genetic approaches. They are then fixed, stained, and imaged on a high-content imager. So that's step 1 all the way through 4. And then this is then followed by image analysis, step 5 here, and then with some data analysis involved. Now, most biologists or cell biologists are very familiar with the markers that's used in the cell painting assay and also the protocol involved in the first half of the workflow. However, many are not trained to handle and analyze the large volumes of data that you generally get from these experiments. So typically, one would have to rely on collaborations with some in-house data science experts to carry out data analytics and then come up with some interpretation of the experiment. And so here, what I want to do today is to present a complete workflow where one can independently complete a cell painting assay from start to finish. We set up a simple cell painting assay to demonstrate how you can carry out the workflow from start to finish. So U2 OS cells were seeded in 380 foil plate, and 11 different compounds were tested at 7 concentrations, and this was done in quadruplicate. This table here shows the dyes that were used and the cellular compartments that they label, and also the filter sets that's used to detect these compartments. Now, I want to draw your attention to the Golgi and actin filament over here. Now, in the standard cell painting assay, the image acquisition for these two structures are captured with the same filter set, that's with Texas red. Now, these two different structures can then be later resolved in the image analysis step if required. However, here we modified the cell painting assay by making use of the Alexa Fluor 750 to label the actin filament instead, and then acquire images using the size 7 filter set. In this way, we can acquire and get more robust analysis of actin that's independent of the Golgi staining. Or we can also, in other words, you can also get more robust analysis of Golgi independent of actin. And we were able to do this using the ImageXpress confocal HT.ai. This instrument uses a laser light source and is capable of imaging up to 8 channels, which makes this imager highly suitable for multiplex or highly multiplex applications such as cell painting. This figure on the right shows the spectral characteristics of the laser light source, and the detection of the different cell compartments are also indicated. So here are images acquired using this modified protocol where we have actin in the captured in the size 7 channel. These are the various organelles or compartments that's labeled with the dyes. So again, I want to draw your attention to Golgi, and that's the actin filament. Now here is an image that was here are images that was acquired using a standard cell painting assay. And so if we were to look at just the actin Golgi channel, you can see the actin filaments quite clearly, but the Golgi punctate or the Golgi structures are a little bit hard to make out. So here are comparison images from the Texas red filter set using the standard cell painting assay and the modified cell painting assay. On the left, you're looking at Golgi and actin that were imaged in the same channel, and on the right, this is just Golgi, and you can see there's a huge dramatic difference between the left and the right. And this is in chloroquine-treated cells because in the treated cells, the increase in number of Golgi punctate is a lot more dramatic. A couple more example images for comparison, and you can see in some cases that, again, with treatment, there is a lot more Golgi punctate. And so this increase in number of Golgi, you can imagine, could be masked by the actin structures if they were both imaged using the same acquisition channel. So this shows that using separate imaging channels for the Golgi and cytoskeleton could increase the sensitivity of the cell painting assay and more robustly represent the cellular phenotypic profiles. The next step in the workflow is image analysis. Here I use the Encarta image analysis software. This software provides measurements that are important for phenotypic profiling, and they include spatial measurements, and this refers to measurements or numbers that refer to the shape, the size, and diameter, etc. It also gives you intensity measurements, texture, and also contextual features such as the spatial distribution of organelles and their relationship to each other. Now to add a little bit here, Encarta also comes with the deep learning-based segmentation tool that allows one to get more robust segmentation that is based on models built with training images. It also comes with some pre-trained models, and one of them is the nuclei model, which I've used to identify and segment the nuclei in the cell painting assay. So shown here in the middle panel is an example of some of the extracted features after image analysis. Here's a screenshot of the software. Again, the image analysis setup is very user-friendly and easy to use. Shown here are example images with a feature mask overlay from the Encarta software. So from here, about 400 87 features were extracted per cell. And so you can imagine that with all of these numbers and from every cell, you would need to be able to have some kind of tool to make sense of all of these numbers. And so with that, in the next part of the workflow, we have to do some data analytics. So measurements from the Encarta software were uploaded into StratoMineR platform. And so when I first started working on cell painting, I was initially stuck at this part over here because I was suddenly faced with so many numbers and I didn't know what to do with that. And then I found the StratoMineR platform, which is a cloud-based data analytics software that you can use for further data analytics. Now, Victor will go into details of the workflow in the later part of this webinar, but very briefly, I will walk you through what I did. So the data was uploaded, and then the features were normalized. And this was followed by data reduction, and in my case over here, I used principal component analysis. The components were then used to calculate the distance score, which is then used for hit selection and cluster analysis. For the next couple of slides, I will just show some highlights of the kind of data or of the kind of output we get from the StratoMineR platform. So one of the steps in the workflow is hit selection, which is very relevant because one of the questions we typically ask in every experiment is whether the treatment effect we give produced any significant effect at all. So in this case, for this experiment, the question we would ask is, which compound treatment gave us a significant phenotypic effect compared to the control? And this can be done with visual inspection. We can pick out compounds that have very strong effects on the cell in this way. So comparing control and the positive control, you can see obviously these two look very different. CCCP treated cells also look very different. However, when we look at rapamycin treated cell, that's when you may stop a little bit and go, I'm not sure if this is significantly different or not. And so with visual inspection, you can use that to pick out cells that show a dramatic difference compared to the control. But then when it comes to more subtle effects, that would be difficult to pick out visually. In addition, just visual inspection is also not a practical approach because when you have hundreds of wells, each with multiple images captured in different channels, it's extremely difficult to go through all of them. And so what is the workaround or what is the solution to this? So prior to hit selection, the data was reduced using principal component analysis. And in hit picking, these calculated factor scores are used to identify significant outliers. And this is being represented as a metric that's referred to as the distance score. The distance score is used to represent the phenotypic effect of treated cells compared to control cells. And shown here is a scatter plot of the distance score for the plate. The color coding here represents the control versus the treated groups. And in red, the red represents the control wells, and the color bars represents the treated group. And so one observation you may see is that there is a large spread in my data points. But this is because each of my compounds were tested in a 7-point dilution series, and so this explains why there is a spread. And because the compounds were used at different concentrations, I was also able to get a dose response curve based on the phenotypic distance score. Hierarchical clustering can also be performed on statistically significant hits based on the selected parameters or calculated factors. Now, clustering would group your cells with similar phenotypic profiles together. Shown here is a dendrogram that represents the hierarchical relationship between all the significant compounds. There are 10 clusters over here, and each are represented by these colored bars here. Now, most clusters are made of one compound treatment. For example, where I'm pointing to right now, these pink bars are all in one cluster, and they're made up of only CCCP treated cells. Next to it is a staurosporine cluster, and then next to the staurosporine cluster in yellow, and this is a cluster that's made up of latranculin. However, there are also some clusters that are made up of different compounds, such as over here, taxol and rotenone, tetandrin chloroquine, cytochalasin D, and latranculin. So what does this mean? So one of the ways clustering can be useful is that you can use it to identify compounds that have similar mechanism of actions. And therefore, for example, if you have a novel compound and you don't know very much about it, and this novel compound is clustered with a group that is made up of compounds that have a specific kinase inhibitor as a mechanism of action, for example, you can use that as information to design future experiments to figure out what the mechanism of action is for this novel compound. And so just to illustrate some example, this first cluster over here is made up of cells that were treated with tetendrin and chloroquine. These two compounds are known to have effects in the autophagy pathway. And although I don't have a specific marker for autophagic vesicles in the cell paint assay, we can see that there are similarities in the phenotypes of these two cells, of these two groups of cells. For example, you can see that there is an increased number of these Golgi punctate vesicles, of the Golgi vesicles. And there's also an increase in the ER channel, which is represented in green over here between these two. Another example I want to show is this cluster that is next to the blue cluster. This green cluster is made up of latranculin and cytochalasin D treated groups. Now, these two compounds are known inhibitors of actin polymerization. And so we can definitely see that there are similarities between these two images. Cytochalasin D and also latranculin is known to inhibit cytokinesis. And so one of the hallmarks of such inhibition of cytokinesis is the presence of multinucleated cells. And you can see this represented in both of these images. And so to sum up, I would like to recap on some of the main points from this part of the presentation. I hope I've convinced you that phenotypic profiling is a simple yet powerful image-based tool that has diverse applications. The workflow that I've shown is easy to implement. You just need a high-content imager, image analysis software, and data analytics software. Now, most high-content imager come with analysis software, or you could use any of the freeware, open source freeware available. And because as a cell biologist by training, the bottleneck for me really was in data analytics, I was very happy to partner with StratoMineR to be able to use that platform to help me interpret all that data I'm getting from my cell painting assay. And last but not least, I would like to acknowledge my following co-workers at Molecular Devices, all for their very useful discussion and help along the way. And of course, as I've mentioned, to Call Life Analytics, to David and Victor, both of whom have been very helpful in answering my many questions and also for many useful discussions. And with that, I'm going to stop here. Thank you all for your time, and I'm going to pass this presentation over to Victor now.
Thank you, Angeline, for the lovely talk covering high-content phenotypic screening using cell printing approaches. And for my part now, I'll continue with that story to cover the questions that you've not considered or perhaps even afraid to ask. And more specifically, I will address the issue of how to work with big data. This talk is to emphasize how to turn Angeline's wonderful images of cells into biologically meaningful information. It will not be heavy on the technical aspects, but rather the emphasis is really more on the fundamentals of analyzing and playing with the data at the end of the workflow that Angeline showed earlier. Essentially, how do we get from acquiring these beautiful images from steps 1 to 5 to the finish line at step 6 to 7 with new, meaningful biological knowledge? When it comes down to high-content screening, the data that you get after image analysis is not quite a simple case of plugging in formulas, reformatting your data as you typically would in Excel or GraphQL Prism, predominantly because of the volume of numerical values that Angeline had extracted from her image analysis software. Her segmentation process extracted 250 different measurements, and this table here of numerical values only shows a small part of the 250 features spanning across the table here and beyond. Going down the columns are the values or objects, and I believe Angeline extracted the features at an object level of resolution. This is why the output can be quite overwhelming in terms of comprehension, but with the right analytic workflow like StratoMineR, you can handle them very rapidly and easily. So how did Angeline get from here with all these numerical values in her data to there with all the wonderful visualizations of the data? So how did we get from A to B, as shown in the previous slide, from numerical values in the dataset to produce biologically relevant knowledge from the dataset? Well, there are key things to keep in mind for a successful multiparametric data analysis, and that is to make sure that you have a consistent analytical workflow. Workflows that goes all over the map deviating from this journey makes it very difficult to reproduce. And this is particularly true if you have everyone in the lab analyzing big data in their own way, using various Python or R packages. But as an example from our StratoMineR data analytics platform, we follow this general workflow that should not only allow accessibility of the data, but should ensure reproducibility as well as transparency of your analysis. Additionally, you must also be able to simplify your data to make phenotypic screening more humanly understandable. I'll go into more detail on this point later. Lastly, but not the least, the analysis workflow must be iterative, so that when new biology is being discovered, of course, there are new questions about. So it is important that your analysis allows you to go back with the new information on hand and reanalyze the data from a new perspective. And in this workflow that I will go over today includes assessing the raw data, eliminating redundancy in your dataset, basically simplifying your data, quality control, removing bias in your dataset, data reduction, and then hit selection and clustering. So let's dive right into this workflow I was talking about in a little bit more detail. First of all, big data output from high-content screening like Angeline's are typically in flat file formats with comma or tab separated files. These are usually large in size that can go up to 10 plus gigabytes with hundreds and hundreds of rows and columns of numerical values, and therefore you won't be able to open these files in Excel or other spreadsheets. While this may seem trivial to you now, but when it comes down to it, this is the first thing that you have to do. So you need to have a platform like StratoMineR that can accept large data files quickly for you to analyze your data. So after when you've uploaded your data, there's still a few things that you need to do. Remember that when I mentioned earlier that it is important you have to simplify your data. And one of the ways to simplify your dataset is to be able to detect good data from the bad data. This means that you need to eliminate features that are deemed unuseful or not useful. And what do we mean by not useful? This means measurements and features that contain non-numeric values, that they are discrete, that there is no variation, there's excessive missing data, there's high mass Pearson correlation value, and that there is uniform distribution in that particular feature. So it is important to check out the garbage and thereby reducing the computational time and energy to carry out the rest of your analysis. Now again, I want to emphasize the consistency in your data analysis workflow as well as detecting these redundant features is key here. So this can then be applied robustly over and over again to remove additional redundancy as you add more and more phenotypic datasets. Now in StratoMineR, we have built precisely that a sequential and logical step that is similar to the list here to detect and to eliminate redundant measurements. So in StratoMineR, it will detect whether or not the data is binary or continuous. Anything that is binary will be considered to be redundant. How many unique data points that you have in the measurement is also another obvious criteria, especially if you have less than 3 unique values, then that feature is probably telling you nothing interesting. And if you have a standard deviation, it's another one that indicates the variation of your features. If it's too low, it will be automatically flagged. And if you have a high McPherson correlation value, meaning that you have 2 or more measurements that are highly correlated with each other, then one or more of these features are considered to be redundant. After all, why would you consider multiple features that measures the same thing? After removing the redundancy in the data, it is also critical to be able to create a plate map for your experiment, where you can assign specific values to your treatment groups, for example, where the negative and positive controls are, and where the compounds of interest are located. This extra layer of metadata information will be useful downstream of your analysis process, where you can track things as you go through the data analysis workflow. This plate map here is associated with Angeline's cell painting experiment, where she labeled her negative and positive controls, as well as some of the compounds that she's interested in, comparing all the other samples with, say, for example, chloroquine or lactranquilin B. But after creating the plate map, this follows then that you'd like to see a quick overview of your data, visually and interactively. And it's important that you are able to visualize to see how your data is being distributed based on the various X and Y axis, and interactively to the point where you can select data points and remove some of the obvious outliers, and or assign a cluster of interesting data points to create a new reagent classes or groups. And this is particularly important for a number of reasons, and this all points to one thing: quality control. And so to elaborate on this a little bit further, quality control is important because it makes absolutely no sense to do high content screening, especially when you're at the assay development stage. And if it takes you many months or even years to analyze your data only to find out at the end that your controllers are not working as they should, for example, then it's a bit too late, because already a vast amount of time and resources have been spent. And so having an analytical workflow that includes quality control built into your process will allow you to rapidly assess the assay. And this is tremendously important, especially seeing how well segregated your negative and positive controls are, and thereby allowing you to calculate something like a C-factor to see how good of an assay you have, or whether your controls are giving the phenotypes as expected. The higher the C-factor, the better the assay is. And this is just one example. There are plenty of mathematical approaches that can allow you to assess the quality of your assay. And when you're dealing with multiple plates in your high content screens, you need to be able to normalize your data. And why is that? Because there is a hidden impact of plate uniformity. As indicated in the assay guidance manual, it states that place-to-place variation in raw signals can induce the appearance of significant midpoint variation, when in fact that there is little variation in signals properly normalized the on-place controls. And the goal of place-to-place normalization is to remove or take into the account of place and edge effects, and to have unbiased comparison of well X versus well Y in place number K. And if you want to know more about how to do a good screen, the Essay Guidance Manual is a great resource and a great place to start. It's developed by the NIH that is practically the Bible for any essay development, including high-content approaches. I urge you to also check out the workshop that is available online and enjoy the great wealth of information on the basics and the good practices to consider. And here is just an example of the 15 plays from one of the phenotypic screens, and you can quickly see that they are batch or place effects. But when you normalize all of that to say the median of the negative control, you effectively remove these place-to-place variations. Then if you are making use of multiple exact features or measurements, however, then there are some mathematical assumptions that need to be met, and that's what data transformation is about. For the math commonly used in phenotypic data analysis, there is the assumption that your measurements or features are normally distributed. If they are positively or negatively skewed, you can transform them back towards a more normal distribution. This is critical to remove bias towards features that are heavily skewed in the negative or positive direction. There is an additional step to remove bias in your dataset, and that is to scale your features. Because measurements can have very different scales or numerical range. Think of measuring length in millimeters versus kilometers. You need to standardize that. In multivariate analysis, measurements that produce large numbers can have more influence than those with small numbers. So that introduces bias in your data set. So you need to essentially level the playing field across all of the features that you've extracted. Data scaling helps to take care of this problem. This is also important for machine learning algorithms that calculate distances between the numbers. And then here is a whole bunch of features from Angeline's dataset. As you can see in the raw data up here, the x-axis are the features she extracted, and the y-axis is the numerical range. Some of the measurements that she has can be up here in the tens of thousands, and this will overwhelm the ones down here. One thing then you can do is to relate every data point to the standard deviation of the distribution for that measurement. This generates what is known as the Z-score. Now you can see how the scales are much more similar. A better way to do this is to use what's known as the robust Z-score. This is less sensitive to extreme outliers. What happens when you have missing data in your dataset? And taking care of missing values is a dirty little secret of data science. If you have a hole in your dataset or your matrix, you can crash many of the algorithms that you'll be using for dimensionality reduction or machine learning. And of course, it's not valid to just put a 0 in there as a substitute for the missing data. For this reason, we try to figure out what values should be there. We're not going into too much details in this webinar, but there are many forms of regression or even machine learning approaches that can be used for this purpose. And in StratoMineR, we have a very simple method that uses the values of other replicates that do have values, or perhaps the median of all the values for that measurement. The end result should never change the overall variation in your dataset for that measurement. And the worst thing that could happen is that you get a false negative for that particular feature. high-content screening often use high multiparametric measurements, and is essential to reduce the dimensions of the data. What this means is that you're compressing the data from a high to low dimensional space without losing the original information. Why this is important is due to the curse of dimensionality, which means that if you have too many features, there is less accuracy for classification purposes. And by performing data reduction, you remove additional redundancy in your dataset. And by having less dimension, it's good for faster analysis and make it more compatible with algorithms that are unsuitable for high-dimensional data. And then you can easily and rapidly access the data by looking at what groups of features are giving you a similar or different story. So by performing data reduction, like principal component analysis or common factor analysis can also yield important insights into which features or measurements sums out the most, phenotypically speaking. And then you can quickly explore the variation of the data in each component or factor. This is extremely helpful in giving biological meaning to non-intrusive features, and therefore this step is critical for basically seeing the biology coming out of your dataset. But after all that's said and done, you can then take your data to find out which compounds or hits give distinct phenotype relative to a specific reagent class, and this can be a group of compounds or a negative control. For example, here's the hit visualization that Angeline has shown earlier, where it's a simple case to find hits from her samples that are phenotypically different from the negative controls. The metric to measure that phenotypic differences is the Euclidean distance score that basically calculates how far are these samples from the negative controls. Okay? And so this helps you identify compounds of interest, as these compounds apparently result in a very different cellular phenotypes. Moreover, you can also find hits that are phenotypically similar to, say, the positive controls as well. So there is a lot of flexibility to play with. And moreover, I won't go into too much details here, but there is an attractive thing about high-content screening and imaging, is that it's very amenable to machine learning and the use of artificial intelligence. And this is already being implemented in StratoMineR to recognize phenotypes of interest. The development of this AI model can then be used to apply to your growing number of images generated, where it can find hits based on the parameters that you set. As a matter of fact, nowadays, deep learning can take the step even further by applying trained models to carry out automated segmentation, feature extraction, and data mining to find the hits of interest. What is also useful to include in your data analysis workflow that can really enrich your knowledge from your data is to be able to take those hits that I've shown you in the previous slide and incorporate additional information about the different concentrations of those hit compounds. You can then generate a dose response curve. This is very informative for any drug discovery pipelines where you can quickly see not only the hits of interest, but also how they elicit phenotypic response in a dose-dependent fashion. And for example here, one of the hits that Angeline had was a CCTP, and StratoMineR can extract concentration information that is in the metadata to generate this IC50 curve based on distance scores. And the images from Angeline's cell painting approach can then be added on to verify and visualize the phenotypic effect, which is extremely obvious. Lastly, but not least, it is useful to be able to take the information from your hit selection step and plot them into a clustering graph. For instance, here again shown by Angeline earlier, to cluster all of her hits using hierarchical clustering, based on phenotypic similarities across all the components she made in the dimensionality reduction step. Here you can obviously see some of the compounds are clustered together that make total sense. But when you also see other compounds being clustered together, for example, chloroquine and tacrolimus, indicating possible similar mechanisms of action to achieve the same phenotypic outcome between the two compounds. So to summarize, when you develop your high-content data analysis workflow, you need to consider these key points here to ensure the robustness of your analysis pipeline. And I cannot emphasize enough that having a robust workflow like we have in StratoMineR that doesn't go all over the place, and it will pay off enormously at the end by keeping your data analysis straight and narrow. And with this workflow, having huge amount of data from high content screens is no longer something to be afraid of or to be intimidated by, because with this workflow now you can make your data and your data analysis accessible, reproducible, simple yet versatile, and transparent. So this is how you can successfully get from A, with beautiful images coming out of high-content imaging instrument, to B, where you can visualize the data and analyze them quickly and intuitively to discover new knowledge from your data. So yeah, thank you very much for your attention, and we hope that the webinar is informative and help answer the questions that you have not asked yet. And more importantly, that we hope that this webinar excites you to use self-painting approaches with robust data analysis workflows to accelerate your scientific discovery pipelines. I'd like to extend my gratitude to my colleagues at Molecular Device, Angeline and Suzanne, and our core life analytics, David and Renan, who are the co-founders of StratoMineR Data Analysis Platform, and Michael Stewart. Now I will hand over the floor to you, the audience. If you have any questions or concerns, please don't be shy. We're looking forward to hear from you. Thank you.
Thank you, Angeline and Victor, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, do you have to use confocal imaging for cell painting, and what about wide field microscopy?
I think I can pick up on that one. You, you can use confocal imaging if you want to, but there are better ways to accelerate the imaging process, because if you use the traditional confocal imaging, then you have to take pictures one at a time. But with the high content imaging instrument that ImageXpress they provided by the molecular devices, you can accelerate that process by adding the automation behind the image acquisition Am I right on that, Angeline?
Yes, you're correct. So you can use either confocal or wide field microscopy to do cell painting. In fact, one of the first papers from Ann Carpenter's lab was done on a wide field imager.
All right, thank you. Next question. Regarding the image analysis, how long does it take to analyze a plate?
Let me answer that question. So for the initial image analysis part of the workflow, in the Encarta software, you can take about 3 hours to analyze a 384 well plate that's imaged with 6 channels and 4 sites per well. The time changes depending on how complex and how many steps you want to add into your analysis protocol. So, for example, if you use a lot of deep learning models, that would take more time.
And regarding the StratoMineR software, that also depends on the granularity of the data. So if it's either captured at the object level, single cell level, field level, or well resolution, usually within an hour, you can analyze hundreds of plates at the well level and at field level, and usually at single cell level, depending on how large the data is. So number of plates and number of features and number of cells per well. It can take up to a few hours if you get into the let's say tens or hundreds of plates.
All right, thank you. Next question. How long does this approach take to analyze an average experiment from beginning to end?
Let's see. From beginning to end, an average experiment. So if we're going to start with the bench work, that would take a few days just to get an experiment set up. And once you acquire the images, and if you're familiar with your workflow already, you can get it done within a day. I would say, maybe Renan or Victor would have something to add on over here.
Yeah, I think.
Go ahead.
Okay, I think it's mostly on the wet lab stuff and on the roboting, pipetting / image analysis stuff. That's the majority of the time that it takes. I think especially on the bench and wet lab stuff. That's to my knowledge the most time intensive tasks that need to be done in order to get through the whole workflow from the beginning to the end. I think the real data analysis part. If you know exactly what you're looking for, of course, then you can done as quickly as a half an hour. But if you're wanting to inspect all kinds of different quality metrics, try out different normalization methods, and act in a very explorative manner, which some of you guys might do, then it of course can keep you busy for weeks if you're not sure what you'd like to find. So that also really depends on the way you're doing your analysis. I hope that helps.
All right, thank you. Next question. What is the learning curve like, and what are some of the skills needed to start using it?
Maybe I can go first since I was completely new to this when I started. You don't, well, at least to use the StratoMineR portion, you don't need any programming skills or knowledge. It was quite easy to get started in my personal experience.
Right.
And Victor, Renan, do you have more to add?
Yeah, I want to add to that in regard to like getting started to use the high content approaches. When I was a postdoc before joining Core Life Analytics, I was involved in actually starting up this whole process of high-content phenotypic screening approach to find compounds of interest for neuroscience. And I must say that the learning curve can be quite steep, but as long as you have the concept and the foundation in terms of knowing which markers and knowing how to validate and optimize your assays. And this is why I strongly recommend anyone who wants to know more about high content screening. Go to the essay guidance manual provided by the NIH. It's a great resource and it's actually what got me started up and running the high content screening core over there. And in regard to the data analysis, yeah, I mean, we developed this platform with that robust workflow so that you don't need to know programming skills. As long as you have a foundation in statistical analysis and things that you would normally do in the lab, then you can do that, and it's nothing to be intimidated by or be afraid of.
Yeah, I think Victor has a very good point there, and I think I'm the worst person in the world that can or cannot say that StratoMineR is easy to use or not, because yeah, I for a large portion developed this software myself in the early days, and yeah, next to that also Victor has been working on a manual that is integrated in the software in the next version. So then it will be really easy for the let's say the step where you are at that moment. So let's say the QC part, then you can pick your brain in the manual and then see exactly what you can do there and how you should be able to do that. And in addition to that, we're also together with David Egan, my co-founder, and some other great researchers were, I think, already busy for the last 3 years of rewriting the assay guidance manual. So there will be a new version released, but I'm not sure when that will happen. But we're also busy on the writing that, and who knows when that when that new version will be published.
Yeah,
We're
I'm not
also
sure on that
contributing
one.
to that.
Yeah.
All right. Thank you. Next question. What are the computer tech requirements for the image and data analysis?
So I can talk about the image analysis part of it. I analyze the data on a standard imaging workstation. It's not any supercomputing device that we have for that. So I would say like it's got a certain number of cores that you need to run analysis, but I think any typical image analysis workstation will work.
And
yeah.
sorry, go ahead.
No, it's okay. I just want to add on top of that is that for this type of data analysis thing, that as long as you have a functional computer in terms of like a working laptop, you can run your own data analysis workflow, or you can use our StratoMineR platform. And and I do want to emphasize that the difference is that without StratoMineR platform, you can do it on the cloud environment. And as long as you have an internet connection, you can access it anywhere. And it's critical in that regard because then, if your lab mate or your colleague is somewhere and they would want to access your analysis, then you'll be able to do that as long as you have internet connection. In regard to working in your own laptop and working in your Python script or R programming, what have you, you should have to make sure that everyone have access to your analysis to see what you've done. And so there's no real specific computational requirements or, or, anything of that kind.
Yeah, I have recently received the recommended specs for running Synapse, for instance. I can share that if you guys are interested in that.
All right, thank you. And it looks like we have time for one more question. In addition to the dyes you mentioned, is the cell painting protocol amendable, compatible with additional markers of interest, say SOX9? If I want to isolate that specific SOX9 positive or SOX9 negative cell populations and compare between the two.
Yes, you can use additional dyes. Again, because at least the imager that we have has eight channels, and so you have room to add on more dyes. But do take note of potential bleed through or spectral overlap in this case. So make sure you optimize your antibody or your dye staining. I mean, you can also instead of adding more dyes, you could also swap out dyes. So the cell painting protocol is very adaptable to your specific needs. Yeah, and then in this case, you could also then in the image analysis step identify cells that have the marker that you're interested in, and I guess you're asking about SOX9, and then just have that two cell populations defined. So yeah.
All right. Well, thank you, Angeline and Victor. Do you have any final comments for our audience?
I don't really have anything except it's been a very good learning journey personally, and that yeah, that's what Victor has said. It's you don't have to be afraid of the data.
Yeah, and I would like to also add that I've forgotten to mention one key term here is that when you're using high content phenotypic approaches, you are actually tackling the biological question with a very holistic view of what is happening within the cell. And that's tremendously important because just remember now when I was back as a graduate student, and my supervisor would always emphasize that you always have to take science in a holistic, integrated, holistic view of everything so that you can better study what is happening in the system. You can't just be very narrow, let's say, for example, just looking at one specific signaling pathway and see how that affects the cell. It would be great if you can look at, other things that are happening within the cell in addition to a compartment that you're interested in. And so I just want everyone to keep that in mind. If you are interested in a more integrated, more holistic view approach to cell biology, this would be a best bet.
All right. Well, thank you again, Angeline and Victor, for your time today. We would also like to thank LabRoots and our sponsor, Molecular Devices, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.